Greetings. I'm Professor Robert Schwartz, Chapter President of Sigma Xi, the Scientific Research Honor Society, established in 1886. Sigma Xi is one of the world's oldest and largest scientific organizations. It's a distinct privilege to welcome all of you to this inaugural Rajendra Kapila lecture. It pays tribute to Professor Rajendra Kapila, an esteemed infectious disease expert, sagacious physician, and beloved educator for more than 50 years here at Rutgers, New Jersey Medical School. Raj passed away 2 years ago. Revolutionary for more than 250 years, Rutgers University is one of 9 surviving colonial colleges. Accordingly, we thank King George III for establishing us in 1766 in honor of his wife, Queen Charlotte. Today, the former Queen's College, now known as Rutgers University, is an academic, healthcare, and research powerhouse. It is in that spirit that we have the tremendous honor to introduce our speaker for this inaugural Rajendra Kapila lecture, the 2020 Nobel Prize laureate, Dr. Harvey Alter. Dr. Alter received his undergraduate medical degrees from the University of Rochester and rapidly rose in academic medicine, joining the National Institutes of Health, where he made groundbreaking contributions. He co-discovered the Australian antigen, pivotal in detecting hepatitis B virus. His work later led to markedly lowered risk of transfusion-associated hepatitis. He was the principal investigator on studies that identified the virus, what was then called non-A, non-B hepatitis, now called hepatitis C. In 2020, Dr. Alter won the Nobel Prize in Physiology or Medicine for his role in the discovery of hepatitis C virus. He is the Distinguished NIH Scientist Emeritus, as well as Senior Scholar at the NIH Clinical Center. Dr. Alter, with extraordinary gratitude, the floor is yours.
Pleasure to be here today, and honored to do the inaugural Kapila lecture. I did not meet Dr. Kapila, but he sounds like a wonderful scientist and clinician. My opening slide, this title is pretty much what I'm going to talk about today. I think we are at the end of the beginning of the hepatitis C story. I'll show you how we got there. And we have to consider whether this is now the beginning of the end. I'm going to tell 2 intertwined stories today. First is a very brief story of my life. I'm telling this because I think it has some lessons for young investigators. And then I will tell the life and possibly the death of the hepatitis C virus. Now, I knew very early on that I wanted to be a doctor, as shown in this picture. But I wanted to be a real doctor. I wanted to have a stethoscope, but I wanted to see patients in practice. That was my goal. It didn't turn out exactly that way, and that's why I think my life is instructive, because you don't quite know where it's going to go when you're just starting out. This is a picture of me at age 13 at my bar mitzvah, and I show that to point out that huge pompadour and the full head of hair, proving once again that I'm not really bald. I'm a hair donor, and I hope you'll keep that in mind. Now, there are 2 critical turning points in my life. This perhaps was the most important. I was at University of Rochester Strong Memorial Hospital in my first year of residency when I got this draft notice telling me to report to Fort Dix, New Jersey, right near you guys, uh, on November 30th. And I'd already applied to NIH, but had not yet been commissioned in the Public Health Service, which was an alternative to the draft. With some frantic calls and some support from the staff, I was able to find that I could get to NIH if I could get commissioned in the Public Health Service prior to my draft notice. I was able to accomplish that, but I point this out, this was life-changing, because had I gone into the Army, I would have been sent to Berlin. There was some Berlin crisis at the time. And then I would have certainly gone into practice as I had always planned. But because I came to NIH, I got into some interesting research, and it sort of redirected my life. We found, while looking for something else, while looking for polymorphisms in the beta lipoprotein system, which Dr. Bloomberg had already discovered, we found a precipitating line which didn't take up the blue stain of the known lipoprotein polymorphisms. It just stained red for protein. Turns out this was a reaction in agar gel between an Australian Aborigine and a multiply transfused hemophiliac who presumably had the antibody while the Aborigine had the antigen. This Australia antigen, so named after the place where we found it, later turned out to be the surface coating of the hepatitis B virus. And Dr. Bloomberg, my mentor at that time, went on to win the Nobel Prize in 1976 for taking this Australia antigen already to the proof that it was the hepatitis B virus surface antigen. This is a picture of the hepatitis B virus, the so-called Dane particle. This is the complete virion, but the surface coating here is the Australia antigen, or hepatitis B surface antigen. This wasn't elucidated until many years later. The second critical turning point for me was I was on the faculty, I was a fellow at Georgetown University Hospital in hematology, and had finished my fellowship and applied for a prestigious practice position in Washington. And to my chagrin, I was rejected for that position. They said they really needed a cardiologist, not a hematologist. And so I didn't go into the practice that I thought I was going to go into. And my boss at Georgetown said, well, why don't you stay here? You can always go into practice. So I got into academic medicine, and then I no longer wanted to go into practice. I liked academia. And then in 1970, I got called back to NIH because this Australia antigen that we had found now was clearly part of the hepatitis B virus. And I was asked to continue studies on this agent. This is a slide I show. It was a congratulatory card I received when I won the Nobel Prize, and I thought it was profound. It said, there is no elevator to success. You have to take the stairs. If there's any message I want to leave the young people with today, it is this. Research, life in general, is a stepwise process. You don't jump to success off the bat. And stairs can go up and they can go down. There'll be disappointments along the way, but you keep treading. I'm going to show you how these steps led to the discovery of hepatitis C virus. But this is the one slide I think you should remember if you're starting your career. And the study we began was a prospective study of open heart surgery patients at the NIH Clinical Center. We chose these patients because they went on a heart-lung machine, which required 7 units of blood just to prime the pump. And they received an average of 17 units per patient. So they were heavily exposed. if any agents were being transmitted by blood. We sampled these patients prospectively, pre-transfusion, and then every week to 2 weeks for the first 12 weeks, and then monthly for the next 3 months. We didn't have much we could do with those samples. We could measure the ALT, the liver enzymes indicative of hepatitis. We had the early test for hepatitis B surface antigen, and that was it. Things like PCR came much, much later. But we put these samples into frozen storage and went back into them multiple times as new technologies advanced. We also saved the donor samples so we could make donor-recipient linkages. So it was these samples that was so important to this elucidation. First thing we found, based on ALT elevations, was that the rates of post-transfusion hepatitis were exceedingly high, exceeding 30%. Most of these cases would not have been recognized had we not been doing a prospective study and measuring liver enzyme elevations, because most of the cases were asymptomatic and would not have come to the attention of the surgeon or physicians. And they would have happened when the patient was already discharged home, and we would have missed it. We went out to their homes and obtained blood samples. We found then that the reason for the high rates of hepatitis was this study done by John Walsh, which showed that if you received at least 1 unit of paid donor blood, so-called commercial blood, you had a 51% chance of developing hepatitis. Whereas if you received only volunteer donor blood, the risk was only 7%. With this knowledge, then in 1970, we stopped using these paid donors, went to an all volunteer donor system. This was difficult to do in those days, but we were able to do it. And we introduced the first generation test for hepatitis B surface antigen. You can see this caused a precipitous fall from over 30% to under 10%, even though the amount of blood transfused remained the same, as shown here, 17.5 units versus 18.2 units. Then in 1973, Abbott Laboratories developed a more sensitive test for hepatitis B surface antigen. We went back into the stored samples and found that only about 25% of our cases, 30% at most, were due to the hepatitis B virus, shown here in purple, and that there was some other non-B entity which was the dominant cause of post-transfusion hepatitis. The only other known virus at that time was the hepatitis A virus, and was only known by epidemiology. But in 1975, Feinstein and Kapila and Purcell, shown here from your right to left, using this immune electron microscope, which is still in the hallway of Building 50 at NIH. Using immune electron microscopy, Steve Feinstein discovered the hepatitis A virus. Now, Steve and Purcell and Kapila were our collaborators, so we immediately sent our non-B samples to Steve, and he tested them and found that not a single one was due to the hepatitis A virus, the only other known hepatitis virus at the time. So it was then, in a brilliant step of deductive reasoning, that we said if these cases were not A and not B, we'd call this the non-A, non-B agent. Now we use that awkward nomenclature because we hadn't yet proven that it was a virus. And if it was, we didn't know how many different agents might be involved. So we did not use hepatitis C at that time and just kept the name non-A, non-B. With great chutzpah, we thought that we would find this virus very rapidly and then rename it hepatitis C. But that did not happen. What did happen is we had a chimp model. Chimps were instrumental in our early studies. We can't use chimps anymore because of, quote, ethical reasons. But the chimps loved being in our studies. You can see them here. They were lining up for their informed consents, and they were very active participants. Some people feel that they should have received the Nobel Prize rather than myself, but who knows? In any case, with the chimps, we were able to transmit hepatitis from our patients who had both acute and chronic non-A, non-B hepatitis, and also importantly from totally asymptomatic blood donors who had been implicated in cases of hepatitis transmission to their recipients. So that was the next step, this chimp model. Next big step was this patient, Mr. Hutchinson. He was enrolled in this prospective study, but before he was enrolled, he was an inveterate mountain climber, and he liked to blaze trails. And one day while out on a mountain, he had a cardiac arrest, and he would have died on that mountain, but his wife was there. She knew CPR, and she brought him back to life, and eventually he made his way to NIH. We have followed Mr. H. We followed Mr. H for 30 years, and he was invaluable in many of the findings that I'll go into. This is his clinical course on the left. Just concentrate now on the blue curve, which is the ALT elevations. You can see that at point 0, he was ALT normal. At week 5, the ALT began to rise and rose precipitously to 2,112, and then came down rather rapidly as well. We were able to obtain an apheresis unit, a plasmapheresis unit at point A on the rising limb of the ALT, and inoculated that into a chimpanzee shown on the upper right panel. The chimp came down with hepatitis, and Bob Purcell then titrated this material in other chimps and found that it had an infectious dose of 1 × 10⁶.5 chimp infectious doses. Later, we took another sample at point B, and this was put into a second chimp. This was the peak of the ALT, and yet it was not infectious. And we think the reason for this was when we could go back much later with PCR, shown here in the yellow line. You see that at the point of transfusion at week 1, right after transfusion, there was a little blip in HCV RNA. This was the donor virus being passively transfused to the recipient. It then seemed to be contained, but then secondarily rose around week 4 and went up rapidly to a titer 3 × 10⁷ genomes, almost identical with the chimp infectious titer. And then the virus came down very rapidly as the patient got better. At point A, he was very sick. He was jaundiced. He felt miserable. He was nauseated, vomiting, and then he got better quite rapidly. We think this point B sample was not infectious because the virus was on this rapid decline at that point, and because antibody had now developed, so the virus would have been immunocomplex and therefore less infectious. That's shown here in the bottom, but we don't need to go into that. But now we had 2 things. We had this proven infectious inoculum, and we had the chimp model. And that allowed us to perturb the inoculum and to see what effect it had on infectivity. In this study that Steve Feinstone led, he treated this H inoculum with chloroform and showed on the bottom here that the chloroform treatment abrogated hepatitis transmission, whereas a sham extraction caused hepatitis in the chimp. So this said that the virus must have essential lipid in its, it must be membraned, and it must have essential lipid in the membrane. We did other studies I'm not going to show you with filters where we filtered the agent through various size filters and then inoculated the filtrate into the chimp to see which filter stopped transmission and therefore size the virus. With these 2 studies, we could ascertain that the non-A, non-B agent must be enveloped, had a presumed core, that it was small, and that the envelope had a central lipid. And this narrowed the possibilities down considerably to these small RNA viruses, then called toga viruses, now alpha and flaviviruses. Or it could have been a hepatitis B-like DNA agent, but we had a lot of evidence that it was not related to hepatitis B. Therefore, it also couldn't be the Delta agent because that can only exist with hepatitis B. So it's either going to be a new class of viruses or one of these small RNA viruses. As it turned out to be, proved to be a flavivirus. Well, we kept trying to find a test for this agent, and we could not do it. I'll show you in the next slide. So we concentrated on the patients themselves. And importantly, with the liver service, the NIH liver service, we initially biopsied 39 patients. Marvin Berman was the lead author on this study. And we found that most of the patients had very mild to moderate inflammation of the liver, what was called chronic active hepatitis or chronic persistent hepatitis at that time. But 10% already had cirrhosis, and 13% had severe chronic active hepatitis, which would have gone on to cirrhosis with more time. Indeed, we did follow-up biopsies then on these patients who had more severe outcomes and some of the others. We found that over time, 30% seemed actually to improve. This is probably just a reading artifact, but 45% had stable histology, but 25% progressed to cirrhosis while under observation. And therefore, in the end, we had 8 patients out of 39 or 20% who developed cirrhosis. And that number has held up over the years. 20 to 30% of patients with hepatitis C will eventuate in cirrhosis and some to liver cell cancer. Importantly, of those 8 patients with cirrhosis, 3 of them died of liver failure. 3 had very severe liver disease when they died of their underlying heart disease. We now knew we had a prevalent infectious agent that not only could cause enzyme elevations, but could lead to progressive fibrosis, cirrhosis, and liver-related death. That made everybody interested now in hepatitis non-A, non-B, knowing that it was a really severe disease, could be. But while this clinical severity became evident, we could not develop a serologic and enzymatic, a radioimmunologic, or even an early molecular test that was specific for non-A, non-B. And this was very frustrating. At that time, I wrote this poem of frustration, which I called, "I Can't See the Forest for the HBAGs." It went, "I think that I shall never see this virus called non-A, non-B, a virus I cannot deliver, and yet I know it's in the liver. A virus that we often blame, but which exists alone by name. No antigen or DNA, little test to mark its way. A virus which in our confusion has forced us into mass collusion to make exist just by exclusion. But is it real or an illusion? Oh, great liver in the sky, show us where and tell us why. Send us thoughts that will inspire us. Let us see this elusive virus. If we don't publish soon, they're going to fire us." Well, that poem, which I presented at a national meeting, seemed to have a big impact because shortly thereafter, Michael Houghton at the Chiron Corporation cloned the non-A, non-B agent using the latest in molecular biology. Remember, this is back in the mid-'80s. Molecular biology was just emerging as a discipline. And Michael took known infectious plasma from chimps and humans, held it down any virus particle, extracted the RNA, and then reverse transcribe that to cDNA, cut that up with restriction enzymes, then importantly use this Phage GT11 expression vector, which would not only carry over any genetic information that was in the original knock-on, but would express any protein that the gene coded for. The phage were then used to infect E. coli. E. coli were grown in agar, and ultimately the agar plate was lysed and onto a filter paper, and presumably any protein that was expressed would have stuck to the filter paper. And then the Chiron group made the assumption that there would be an antibody in patients who had recovered from non-A, non-B, or patients who had chronic non-A, non-B, even though no antibody had ever been demonstrated to that point. The story goes that over about 4 or 5 years, they screened 6 million clones before they found a single clone that was positive. But then they could take that clone and subclone it, extract protein, walk the genome and make a bigger piece of protein, and ultimately develop an antibody assay to this virus, which they then called the hepatitis C virus. This is a picture of Michael Houghton on the right at the time he received the Lasker Award. And then later he shared the Nobel Prize with me and Charlie Rice. On the left is Dan Bradley from the CDC, who was also very important in these early studies. Now, when Chiron found what they thought was a specific test for the hepatitis C virus, they asked for a panel that I had developed, so-called non-A, non-B panel. And I sent that panel to them. Now, previously, 19 other laboratories had asked for this panel, and none of them had been able to break the code. It was a small panel, but it was tricky in that every sample, every positive sample was proven infectious in the chimp, and every sample was there in duplicate, and every duplicate was in a random position. Conversely, the negative controls came from donors who had donated more than 10 times and never been implicated in case of hepatitis transmission. And every one of those samples was also in duplicate and randomly distributed. Well, Chiron broke the code perfectly. They detected 3 out of 3 patients with chronic non-A, non-B in 6 different samples in random positions. They detected 2 donors who had been implicated in hepatitis transmission in 4 different positions. They did not detect 2 patients with acute non-A, non-B because they were looking for antibody, but subsequently, all of those patients, those 2 patients seroconverted. And then they did not find any of the 7 negative controls in 14 different duplicate samples. So they had 100% in breaking the code, whereas nobody else had ever done that. We then looked at 15 of our non-A, non-B cases and found that every one was antibody negative pre-transfusion and then became antibody positive 6 to 8 weeks post-transfusion. So they were all hepatitis C cases. And then we looked for the blood donors to 25 of these cases. We found a positive blood donor in 80% by a first-generation assay, 88% with a second-generation assay. So we could presume that if we introduce this test to blood donor screening, we might prevent 90% of post-transfusion hepatitis. Indeed, that's what happened. I showed you the left side of this slide earlier. Over the years, we tried different interventions to lower the rate. The rate did come down because the amount of blood transfused came down from 17 units to 7 to 8 units per case. But when we introduced ALT screening of donors, it didn't have any impact. Anti-HIV testing didn't seem to have any impact. We thought it might be a surrogate. In 1987, based on some retrospective studies we and other groups did, we predicted that anti-hepatitis B core testing would serve as a surrogate for non-A, non-B because of similar exposures. And indeed, that lowered the rates by about 30% as predicted. They came down to 1.1%. And in 1992, a second generation test came along. The rates came down to 0.3%. And by 1997, we were down to virtual zero. We could estimate from the various incidences over the years that blood transfusion may have transmitted 4.8 million HCV infections from 1970 to 1990, those 2 decades. Now, those infections would not all lead to cirrhosis, but there was a 20% chance that some of these patients would develop cirrhosis. Many of the patients would die before they actually got the cirrhosis. But at any rate, we set them up for developing progressive liver fibrosis. Now, conversely, by introducing the test in 1990, we could calculate that we would have prevented 2.4 million transmissions over the next 2 decades, from 1990 to 2010. Indeed, we've now followed about 1,000 more patients for hepatitis B DNA and hepatitis C RNA, and have not had a single case since 1930s. Now, this is that same slide, except it has this big downward arrow. And I show this because this is not only the story of hepatitis, post-transfusion hepatitis, but this is the story of my life. And you can see that both post-transfusion hepatitis and my life are on this very rapid decline. And I'm just pleased, thrilled, in fact, that hepatitis has reached bottom, and I'm still here talking about it. Throughout all this time, I was a government employee, as I still am, part-time. And government employees get a bad rap for being lazy and just living off the government dole. But I want you to know that I always gave 100% at work. It's just that it had this peculiar distribution. So today is Friday. This is really one of my really bad days. So that's the transfusion story and the discovery of hepatitis C. Now I want to talk about hepatitis C as a global problem. We think there are 70 million people now who are chronically infected with hepatitis C, 5 million of those in the US. And these high rates are because 75 to 85% of people who get infected with hepatitis C develop persistent infection. So over the years, we were interested in, wow, what's the reason for this, our inability to get rid of the hepatitis C virus? Working with Patricia Farsi, who's still my main collaborator, she looked at 105 clones from the same patient, Mr. H. Found that 57% of the clones were identical. This was the dominant strain, but there were already 19 other clones already there. So even if somebody developed a good immune response against a dominant clone, any other one could come out, emerge, and become the strain, the dominant strain, and keep the infection going. This is a so-called quasi-species nature of hepatitis C. It doesn't exist as a single variant. It exists as a family of variants, all simultaneously present. The same is true for HIV. To show how this works, this is a patient we looked at who had severe progressive chronic hepatitis C and tested samples at weeks 3, 8, 13, and 16 along the way to the peak of the ALT as shown in blue. Patricia did 10 clones at each point. You see that at week 3, there were sort of 3 co-dominant clones, arbitrarily labeled A, B, and C. But that by week 8, now D was the dominant strain, but there was also E, F, and G out of these 10. By week 13, You can see A was still there, but now there's HIJKLMNOP. And finally, by week 16, all the way through the alphabet to U. The big change between week 8 and week 13 was that antibody had developed by that time. It was putting increased immune pressure on the envelope of the virus and causing increased variation. Overall, when we looked at our post-transfusion cases, we found that most of them had this stable or very slowly progressive course, where they would probably go their whole life without getting severe fibrosis, or it would take 40, 50, 60 years to happen. But 20 to 30%, as I said, had severe and progressive disease where they developed cirrhosis in 15 to 40 years. And a small number, less than 5%, could get cirrhosis very rapidly in 5 to 10 years. We now think in retrospect that these were people who had coexistent conditions, probably either unadmitted alcoholism or we now know fatty liver and NASH, which can lead to cirrhosis even in the absence of a hepatitis virus. Put together, they lead to cirrhosis more rapidly. So we wanted to look then at what's the difference between slow progressors and rapid progressors. Well, if you look at the yellow line, the slow progressors had the acute infection, big rise in HCV RNA, but then contained that infection, came back, almost disappeared, but then came out again as a new variant and then persisted after that. In contrast, the rapid progressors never had that early immune containment. They had high levels of virus from the beginning throughout their course. We also found that rapid progressors had high levels of MCP1, monocyte chemotactic protein. This is a pro-fibrogenic chemokine that attracts stellate cells and then is produced by stellate cells, setting up the cycle of MCP1 stimulating the stellate cell making collagen, leading to fibrosis, the stellate cell refeeding back and making more MCP1. So we think this is one of the main reasons for this more rapid progression. In contrast, the slow progressors had increased levels of interferon gamma and MIP1 beta, and these are the kind of the good cytokines that are anti-inflammatory. And throughout this time, I was not only a government worker, but I was married. And my wife once said to me, "You are the answer to my prayers." But then she went on to say, "You are not what I prayed for, but apparently you are the answer." And that's where I am, marriage. I want to talk a little bit about cirrhosis and liver cancer. This is a picture of a cirrhotic liver with multiple foci of hepatocellular carcinoma. But we looked at this in several different studies. This was a study that's not well known, but was done by Dr. Kendo Kiyosawa in Matsumoto, Japan. And he did this intensive follow-up of patients who were blood transfused. It's complicated, but not so. If you follow this, so the red thing at the end here is the blood transfusion. Then they had their acute hepatitis by enzymes or clinically. And then he had serial biopsies, following them through different stages of chronic persistent hepatitis, chronic active hepatitis, finally to cirrhosis, and then to hepatocellular carcinoma. This is the best documented progression of these things. We went back then and tested these for hepatitis C, showed that all these cases were hepatitis C related. And I want to point out also the duration between the transfusion and the development of hepatocellular carcinoma. Here, near 30 years, a little over 20 years, some short, little as 5 to 10 years, but some as long as 60 to 70 years. So I think this is a very graphic picture of what can happen in hepatitis C. We did another study in Japan that was very instructive, where Dr. Yasuhito Tanaka and Dr. Mizukami, who were experts in so-called molecular clock analyses. Molecular clock is where you can take 2 or more samples from the same patient, look at sequence changes, and extrapolate backwards to the point where this particular variant, this particular genotype, emerged from some common ancestor, and then when it spread rapidly within a population. You see that in Japan, genotype 1b diverged in 1880, and then was dormant in the population until the 1930s and 1940s, when it began to spread rapidly. And then ultimately it has leveled off at this point. Well, why was it spreading rapidly in Japan? Because Japan was in the 1930s, was at war with China, and then later at war with the Allies. We learn in retrospect that the Japanese soldiers, which was almost the entire male population, were often shooting up with amphetamines before battle and then sharing needles. This is how they got such high rates of hepatitis C. Those rates are now tapering off in Japan because the originally infected people are now mostly deceased, and because the young people are not using drugs, sharing needles the way we do in our country. In the US, genotype 1A first emerged in 1910, then began to spread in the 60s and 70s and 80s. And when we had our own needle sharing epidemic, mostly with cocaine, but also with heroin. And that continues unabated to this day. The importance of this slide in the current context is that there's about a 30 to 40 year spread between the Japan curve and the US curve. And because Japan has an 8 to 10-fold higher incidence of hepatocellular carcinoma than we do in the US. And they've always said that you have the virus as long as we do, you will have these very high rates as well. Well, that has proven to be the case. In this study by El Sarag at Baylor, which I had nothing to do with, but he studied the age-adjusted rates of hepatocellular carcinoma in the late 70s versus the late 90s, and found a 3-fold increase in the rates. And it was all due to hepatitis C, shown on the right, not to B or other causes. So it was said then, back in 2009, that assuming there were no changes in the standard of care, the total number of patients with advanced liver disease and hepatocellular carcinoma by 2029 would be 4-fold higher than it was in 2009. And that definitely would have been the case. It would have increased from here another 4-fold. But there was a major change in the standard of care because these amazing drugs came along, these so-called direct-acting antivirals for treating hepatitis C. They were developed in 2011, and we can now cure 98 to 100% of patients with an 8 to 12-week course of oral therapy, which has virtually no side effects. These are really miracle drugs based on specific inhibitors of HCV replication cycle. So that is the end of the beginning. The discovery of non-A/non-B, the eradication of post-transfusion hepatitis, the elucidation that it could be a fatal disease causing cirrhosis and hepatocellular carcinoma, and these miracle drugs which can cure virtually every patient who has hepatitis C. So with this, one could ask, could this be the beginning of the end? And that depends on whether you see the glass as half full or half empty. If you see the glass is half full, you can say, with cure rates approaching 100%, from this time forward, once you identify hepatitis C infection, no one should develop cirrhosis, excuse me, or die of hepatitis C. Excuse me. But if you see the glass is half empty, you know there are hurdles to eradication. Main one is that only about 20 to 40% of people who are presumed to be infected with hepatitis C have actually been identified. And secondly, access to treatment has been difficult, even among known carriers. Only a minority have gotten treated because these direct-acting antivirals at first were very expensive, and few could afford it, and insurance wasn't covering it. But the proportion being treated now has increased because drug companies have realized that it's cheaper to treat the infection early on than it is to treat cirrhosis, liver cancer, and to do liver transplants later on. But cost of drugs has been really the main impediment. They say it's getting better, but we're no longer constrained by science. It's really a matter of dollars and implementation, as I'll get into. So these are the reasons why HCV could be eliminated. Those are the actual numbers, but there's no non-human reservoir of hepatitis. It's not coming from mosquitoes, it's not coming from bats, it's not coming from monkeys any longer. So you only get it by human to human blood transmission. A little bit, it can be spread by mother to infant, but that's a small rate. And sexually transmission is also very low. We have very sensitive diagnostic assays, and we have these curative treatments. So if you have a great assay and a great treatment, it's not coming anywhere else, but from other humans, you can eradicate it. To prove that is a study done in Egypt, where Egypt decided to treat their population. They felt a moral obligation to treat the population. They got permission to make the drugs cheaply in-house and to make tests cheaply. And the results were amazing. So this is the hepatitis C iceberg, where many of the cases are underwater, unrecognized. But Egypt attacked this group of available cases. They tested greater than 60 million people by both antibody and HCV RNA. Those who were antibody positive tested for HCV RNA. About 4% were RNA positive. They then treated over 2 million patients between 2014 and 2018. They had a sustained virologic response, which is tantamount to cure. In 98.6% of those, they could then go back and retreat the failures. They only have, it cost them about $50 to $120 per cure. That includes the cost of the drugs, the cost of testing, and the cost of the PCR, and the most expensive cost of setting up the infrastructure to test that great number of people. They only have about 350,000 people still to test, which they're in the process of doing, and they're continuing to monitor this. Egypt has also offered to provide these cheaper drugs to other countries in Africa. So what we need to do now is this massive global screening, whether it be by country, by state, by community, to immediately or as soon as possible deliver these direct-acting antivirals. They're so safe, you can send the patient home with them and don't need to even follow them until the end. So what we need really to make this work is the political, the corporate, and the moral will to make it happen. So it's not going to happen for me because I'm now in my golden years. These golden years are not really so golden. Age has a lot of downsides. Just for example, this is a picture of Superman as he aged. You see, not so well. Here's Wonder Woman, the same thing. So this depending on younger people now. And it's given me a new philosophy where I have plotted age against potential. And this is me out here. I have used up my potential, but you, Sigma Chi, AOA, and others are over here. You have all this potential ahead of you. You have the chance now to explore other things. Some of you bring HCV to elimination, others to explore other avenues of research. It's exciting. I tell you, if you go in to this kind of life, it's difficult, but it's exciting. It's rewarding. So I highly recommend that some of you at least progress to a career in clinical or basic research. So I want to leave you with this last slide, which is that advise you to show wonderment in the world as you're going in and you're fulfilling your potential. I love this slide of the hedgehog and the young child's concentration on the hedgehog. So be observant of what's going on around you, whether that be the world in general, an experiment in the lab. Observe it and think of the possibilities. Think of where you can go with that observation. And take wonder in it, because it really is an exciting way to go with your life. So remember this hedgehog. If things get tough sometime and the steps are going down instead of up, remember the hedgehog and remember your wonderment, remember the possibilities. I wish you all success. Thank you very much.
Thank you, Harv. Wonderful, wonderful. I'm in wonderment. This is truly amazing. We are extraordinarily grateful. Thank you so much. Thank you. You've had a very, very productive Friday as far as we're concerned. This is for us the penultimate. And so thank you. Thank you.
Thank you, Robert. I can take questions if you want, but
let me see here I'm not sure how to handle that one.
Okay, well, we cut it off.
Oh, well, let me see. Looks like we're yeah, that's it for that one. But it was wonderful. We're so really
wait, Stanley Weiss raised his hand.
Okay, well then why don't you answer? Stan is he's our faculty president. He's a distinguished colleague and friend, and also a co-leader of the Honor Society. Stan, the floor is yours.
Okay, thank you for unmuting me. A fantastic career and a lecture and overview, and in many ways so optimistic for us on where potential may go. I have one question that's mystified me and so many people, and as epidemiologists, in trying to ascertain how some of the people with hepatitis C virus got infected. The major routes, you've well identified, as well as others, but a substantial number of people, we haven't been clear on how they acquired the hepatitis C in the United States. And I was wondering if you wanted to speculate a little bit more about that difficulty of ascertainment and what you think may be going on and how that might affect our ability to further interdict transmission.
Yeah, that's a very good question. Thank you, Stan. When we did we followed the blood donors that we after we had the test, we suddenly realized we're going to have a lot of positive blood donors, which indeed we did. We put them into a prospective study, and in the vast majority had either had a blood transfusion or mostly had used drugs and shared needles back in the 60s and 70s. These were the baby boomers. So we had a lot of people who were not addicts, but they had definitely used recreational drugs at one point. It only takes one shared needle to transmit this. So that accounts for the majority. But I think others came from medical practices, from doctors using improperly sterilized needles, dentists, the same thing, from multi-use vials that got contaminated and used in many patients. This is hard to document, but I feel pretty confident that it's true. If you go back to the 30s, 40s, 50s, the sterilization techniques were very poor, and instruments were just barely washed in the dentist office. Tattooing probably transmitted some in the days when tattoo needles were reused and improperly sterilized. A small percentage may have come from sexual transmission, but this is a very difficult virus to transmit sexually. And when we followed our chronic carriers, and their partners, we didn't find any evidence of sexual transmission. But early in the infection, it's quite possible that one could, during the acute phase and the high viral loads, one could transmit sexually or perinatally. Perinatally transmission is thought to be under 5%, but probably does exist. And we have one case in our study where we thought that was the main route. So those are the known routes, and who knows else. It doesn't seem to be a vector. It doesn't seem to be mosquito-borne. You'd think it would be, but it doesn't seem to be. So that's all. That's the only things I know.
Okay, I'm still in wonderment. Thank you. We're so grateful. Stan, thank you for the excellent question too. And
okay, well, thank you, Bob. I appreciate it.
thank you. Thank you. We're all great. Family's grateful for Dr. Kapila, his widow, Dr. Kapila here, and then we're so grateful. Thank you so much.
Okay, bye-bye now. Bye.